To function properly, the many cells of the heart have to be coordinated in a particular way. For example, the different chambers of the heart have to be activated in a particular sequence, and once activated, all the cells of the heart chamber must contract together. Let's see how they do that. The attachment between two cardiac muscle cells is more than simply an abutment of two cells together. It's an important part of the functional coordination of multiple cardiac muscle cells. Let's magnify this junction for a closer look. The junction between two cardiac cells is known as an intercalated disc. This is a point of mechanical attachment, but it's also a communications link between cells. The intercalated disc is decorated with numerous gap junctions. We've already seen gap junctions in the structure of the endothelial cell. The function of the gap junction, remember, is to allow the passage of small molecules like ions from cell to cell. This means that the electrical activity in one cell, like an action potential, can be transferred through the gap junction to an adjacent cell. And this makes for what is called a functional syncytium. Even though each cardiac cell is mononucleate, their coordination through gap junctions makes them function together as if they were one. The peculiarities of excitation impose a constraint on this mutual excitation. Imagine we have two isolated cardiac pacemaker cells that each contract according to some intrinsic rhythm. On the left, we have a pacemaker cell that has a steep pacemaker potential. Therefore, its intrinsic rate of excitation is fast. On the right is another pacemaker cell that has a shallow pacemaker potential. Therefore, its intrinsic rate of contraction is slow. If we now allow these two cells to couple together through an intercalated disc, the membrane voltages will now be coupled to one another. Whichever cell gets to its threshold voltage first will elicit an action potential, which will then spread to whatever adjacent cells it's coupled to. If a fast pacemaker cell is coupled to a slow pacemaker cell, the slow cell will now be driven by the faster of the pair. This is a general rule of pacemaker cells. In any coupled association of pacemaker cells, the association will beat at a rate that is driven by the cells with the fastest intrinsic rates. Within hearts, pacemaker cells are organized into specific associations known as nodes. Within the vertebrate heart, there are typically two. Near the junction of the sinus venosus and the atrium is the sinoatrial, or SA node. And near the junction of the atrium and ventricle is another node, the atrioventricular, or AV node. In higher vertebrates, there's only one ventricular node, but in the hearts of lower vertebrates like fish or reptiles, the situation is a bit more flexible. Remember that it's only the activation of a single gene that converts a contractile cardiac muscle cell into a pacemaker cell. Within the fish heart, for example, this allows pacemaker nodes to arise spontaneously and transiently as the situation demands. We'll have more to say about that when we talk about the comparative physiology of the vertebrate heart. In addition to the nodes, pacemaker cells assemble into a signal distribution system known as the Cardiac Conduction System. Within the atrium, the conduction system consists of a loose network of conduction cells that permeate as a web throughout the atrium. Within the ventricle, the conduction system emerges from the AV node as a thick bundle, which branches into finer bundles through the ventricular muscle. The nodes and conduction system work together to coordinate the beating of the heart. Most important is to control the phase of contraction of the two chambers. Remember that the atrial cardiac cycle is out of phase with the ventricles. The atrium needs to contract first, and then while the atrium is in diastole, the ventricle has to be in systole. This comes about because the sinoatrial node consists of cells that have a faster intrinsic rate of excitation compared to the pacemaker cells in the atrioventricular node. Because it's the cells with the faster intrinsic rate that drive all the pacemaker cells, it's the SA node that drives the entire system. For this reason, the SA node is sometimes called the cardiac pacemaker. This is a bit of a misnomer because they're all pacemaker cells after all. But because the cells in the SA node have a faster intrinsic rate of excitation than the others, that's the pacemaker that drives all the other pacemakers within the heart. The organization of the conduction system also plays a significant role in coordinating the heartbeat. The network-like structure of the atrial conduction system means that excitation spreads radially as a wave from the SA node, and this takes time. Consequently, there's a time delay in the spread of excitation from the SA to the AV node. Once the AV node is activated, the bundle-like nature of the conduction system there produces a very rapid spread of excitation from the node throughout the ventricle. The overall effect of this is a near simultaneous excitation of all the ventricle's contractile cells. Because the vertebrate heart is pacemaker-driven, it is therefore autorhythmic. That is to say, the heart will beat on its own and in a coordinated way. The heart cells themselves can generate their own intrinsic beats, and by coupling them together through gap junctions, this autorhythmic behavior can extend to the entire heart. By coordinating the spread of excitation through the conducting system, the cardiac cycles of the different parts of the heart can be coordinated. The autorhythmic behavior of the heart can also be influenced by outside sources. We're all familiar with the variation of our own heartbeat, for example, and we'll be talking about some of those as we go along. For now, let's just talk about how the nervous system influences the beat of the vertebrate heart. The vertebrate central nervous system comprises several divisions, and we'll talk about those in more detail when we get into the nervous system. For now, we just want to focus on one of them, the so-called autonomic nervous system. The autonomic nervous system controls the so-called vegetative functions of the body, things like the digestive system, respiration, and circulation. The autonomic nervous system itself is divided into three divisions: the enteric, sympathetic, and the parasympathetic. Of the three, the enteric system controls the function of the gut, and it's embedded entirely within the digestive tract. This will not be relevant to us today, but we'll come back to it when we talk later about the digestive system. The other two autonomic divisions, the sympathetic and the parasympathetic, arise directly from slightly different parts of the brain. Each employ different neurotransmitters to convey signals from the brain to the various organs of the body, the heart included. In the case of the sympathetic nervous system, it is adrenaline or noradrenaline that is the neurotransmitter, and so the sympathetic nervous system is said to be adrenergic. The parasympathetic system, in contrast, uses acetylcholine as its neurotransmitter, and therefore the parasympathetic system is said to be cholinergic. The principal parasympathetic nerve is the vagus nerve, which is the 10th cranial nerve. This nerve arises from around the medulla, and it extends throughout the body, sending out branches to the various organs of the body that it controls. One of its branches extends directly to the sinoatrial node. The sympathetic nerves arise from lower in the spinal cord and extend throughout the body in large paravertebral nerves. These also send out branches to the various parts of the body, including the sinoatrial node and the heart muscle itself. These two divisions of the autonomic nervous system control the heart by modulating the heart's intrinsic rhythmic contractility. As we've seen, the heart can function at some autonomous and intrinsic level, which we'll represent by this horizontal yellow line. Activity of the parasympathetic nervous system acts to slow this intrinsic rate, and the action of the sympathetic nervous system will be the opposite. Heart rate will be elevated. Unlike the parasympathetic system, increased sympathetic input to the heart will also boost the strength of contraction of the cardiac muscle. This introduces the important concept of autonomic tone. The actual function of the heart depends upon a balance between the input from the parasympathetic system and the sympathetic system. Autonomic tone will be the balance between these opposing effects. If the sympathetic system input dominates, heart rate will be elevated and the heart will contract more strongly. On the other hand, if the parasympathetic input dominates, the heart rate slows. It's not so much the activity of one or the other that drives a function of the heart, but the balance between the two. If, for example, the vagus nerve is cut, removing the input of the parasympathetic nervous system altogether, heart rate will increase. Okay, we've learned some important things about how heartbeat is controlled intrinsically and a little bit about how the nervous system modulates this control. The heart is incorporated into the broader circulatory system, of course, and we now have to turn to an important principle that governs that.